Again, that organochip or organoids will not replace or reduce traditional in vitro testing, in regulatory approval of biomaterials, and drugs in foreseeable future. So, my colleague Laura has already presented 4 key arguments: higher cost and lower scalability than existing in vitro tests, extremely slow process for validation and demonstration of superiority or equivalence of existing medical devices. In next 5 minutes or so, I'll be showing you some of the compelling scientific evidences to essentially convince you that organochip or organoids will have limited capacity to mimic complex physiology and in fact unable to replace in vivo tests. They are unable to replicate chronic temporal responses. These scientific evidences are based on our research on biomaterial devices or some of the neuronal chip or bone on chip, as well as some of the ongoing work carried out in my colleagues in India. So, one thing that I would like to bring your attention to, that organochip essentially have inherent engineering limitations of mimicking physiological complexities. Some of the terms which are very used in scientific literature, that microcell co-culture analogs or body on a chip, they essentially fail to recreate physiological complexity, primarily because there is essentially lack of interaction among tissue interfaces and also lack of simulation of physiologically complex microenvironment. So let's take the first example, placenta on chip. This is an ongoing collaboration between IIT Bombay and Indian Council for Medical Research. So placenta is a dynamic, placenta changes structurally and functionally during pregnancy, and the 2 cell lines which are very important with the BBO. So BBO cell lines and one is the Quebec cell lines. So essentially they have grown both the cells in this placenta on chip and trying to find out that whether they essentially produce critical barrier function towards specific drugs and other biomolecules. They have done this high molecular dextran as well as the glucose transport, but major limitations here again is the non-availability of relevant primary cells and also the belief that lack of transport of drug molecules does not ensure safety. So these are the 2 aspects, and in addition to that, regulatory and ethical concerns are, as usual, one of the biggest hurdles in getting this placenta on chip, the due regulatory approval, at least in India. Second one, this is from our own work. A few years ago, one of my PhD students has done neurogenesis on chip. So they have done these biomarker predictive devices. They have done the trans-differentiations of these muscle cells as well as HMSC. So they have done this co-culture. We have given this electric field stimulation in a very controlled manner, and we have shown that this kind of co-culture essentially getting differentiated to neural-like cells. And this work was published in Biomaterials. But however, fully functional neurons cannot be regenerated in this neuronal chip device. The second one, again from our own group, that is osteogenesis on chip. We have grown human mesenchymal stem cells under different shear stresses. We allowed them to differentiate to bone cells, and then we have also seen that a lot of key morphological or phenotypical changes in terms of chirality and differentiation process. But the fundamental process of bone remodeling or stress shielding cannot be unfortunately replicated in this organ chip device. So what are the major difficulties in the organ chip? That dynamic changes over months cannot be simulated, and also phenotypic changes like modifications of shape, rate of growth, those things also cannot be simulated. So I'll give you some more examples in the next slide. So this is the animal studies essentially allow the holistic outcome, particularly foreign body response, and also in terms of the data we have observed that this data is less dispersed with more reproducibility. This is kind of cartoons essentially showing that what goes on during the animal testing, and many of you are very familiar who does this animal testing in their own research. So let's move on to this next example. This is again our own work, which is published in Biomaterials very recently. So here we have used some of the polyhydrogen fluoride with carbon nanotube and also barium titanate. We have used different compositions just to show in the subcutaneous implantations, it essentially induces less of pulmonary response and indeed generates a neovascularization. We have also done in vitro tests, for example, trying to see that what is the hemolysis, and in this particular case, hemolysis percentage is certainly less than 3%, which is quite acceptable clinically. This is the last example, again from our own study, where we have done the medical devices, particularly for dental implants. We have gone from the design of the new design, which is being patented. US patent is due, Indian patent is granted. We have from design to preclinical studies to human clinical studies, all being done in India. And what we have introduced is that essentially one of the specific design that is called hybrid thread design. We have introduced this hybrid thread design to get to exploit some of the clinical advantages of this hybrid thread design. After all, we have done this rapid study after 12 weeks, ISO 10993 guidelines, as Laura has mentioned in her talk, and then we have done a human clinical study on 25 patients. In the preclinical study, we have demonstrated that these specific implants have bone-to-implant contact length at least 20% better than some of the Straumann implants which are clinically used. Now, we have validated our preclinical studies in the human clinical study. In the post-operatively, we have shown that our implants again are superior compared to the Straumann implants. So this validation of the preclinical studies in the clinical trials is something we cannot do, or we cannot get it done in this organ-on-chip and organoids. Is 20% better than some of the Straumann implants which are clinically used. Now, we have validated our preclinical studies in the human clinical study. In the post-operatively, we have shown that our implants again are superior compared to the Straumann implants. So this validation of the preclinical studies in the clinical trials is something we cannot do, or we cannot get it done in this organ-on-chip and organoids. So I am convinced, and I am sure you all are convinced, that organ-on-chip and organoids cannot replace in vitro cell culture. It cannot recreate human physiological system and tissue interfaces. It cannot replace animal models simply because it does not provide clinically relevant insights into spatial temporal responses, particularly vascularization, angiogenesis, and fibrosis. While we prepare these particular presentations, I have done a lot of insightful discussion with my colleagues both in India as well as the United States. I acknowledge those discussions, and also Alpine Biosciences Bangalore, one of the company's startups in Bangalore. Thank you very much for your attention.